Hello dear students. Today we will continue our lecture about the structure of the cell, and on our previous lecture we told that there are two main types of the cell. It is a prokaryotic and eukaryotic cell. And today we will talk about the structure of the eukaryotic cell. I remind you that the eukaryotic cell are the cell of the animal, plants, and fungi. So let's start. There are three main parts of each eukaryotic cell. It is a cytoplasmic apparatus, membrane apparatus, and nuclear apparatus. Each apparatus consists of some parts. For example, cytoplasmic apparatus include cytoplasm, organelle, or another name organoids, and cell inclusion. The membrane apparatus include physiological plasma membrane and all parts of the cell which are located outer, so this part is called outer complex. And also membrane apparatus include all parts of the cells which are located inner of the plasma membrane, and this part of the cell called inner complex. And the last part of the eukaryotic cell is called nuclear apparatus, and this apparatus includes nucleus, nuclear plasm, nucleolus, and nuclear envelope. So today we will talk about cytoplasmic apparatus, about cytoplasm, organelles, classification of the organelles, and about cell inclusion. About the membrane apparatus will be our next lecture, and after that we will talk about nuclear apparatus. Don't remind about this main scheme or main structure of the eukaryotic cell. So I remind you three main parts: cytoplasmic apparatus, membrane apparatus, and nuclear apparatus. And today we talk about cytoplasmic apparatus. Let's start. So we said that the cytoplasmic apparatus includes cytoplasm. What is the cytoplasm? Cytoplasm is the material within a living cell, excluding the cell nucleus. So look on the picture. Cytoplasm is the material within a living cell, excluding the nucleus. It is a complex mixture of dissolved molecules such as proteins, fats, carbohydrates, and water. We must say that the cytoplasm has some cell-cell interactions. Structure of the cytoplasm behaves at times like a disordered colloidal solution, or sol, and at other times like an integrated network forming a solid mass, or it's called gap. Phases cell health is determined by processes polymerization or depolymerization. I remind you that the polymerization is process in which relatively small molecules called monomers combine chemically to produce a very large chain-like or network molecule called a polymer. So it's process called polymerization. The monomer molecules may be all alike, or they may represent 2, 3, or more different compounds. And depolymerization is a waste process. So, phases cell health determine the active or cell biochemical reaction. Phases cell health determine the active or cell biochemical reaction. So, processes are more active in a soul and slower in a health. Uh, Now we will talk about second part of the cytoplasmic apparatus, which called cell organelles or organoids. So what does mean organelle? Organelle is a specialized subunit within a cell that has a specific function in which their function is vital for the cell to live. And now let's talk about classification of the organelles. There are different types of classification, and first classification based on structure of the organelles. Does they have membrane or not? And on the base of these features, organoids divide to membrane organelles and non-membrane organelles. Second classification based on function of the organelles. Does they have special functions or not? And on the base of these features, organelles divided on the special pupils and general pupils. Special pupils such as cilia, flagella, microvilli, microfibrils. All eukaryotic cells have general pupils organelles, but special pupils organelles have only some type of eukaryotic cells with some special function. For example, spermatozoon, male reproductive cell, has flagellum, which help him to move it. Now let's talk about this classification in detail. So, classification of organelles which based on the structure of the organelles. We said that organelles divided into two main groups, membrane organelles and non-membrane organelles. What type of organelles includes membrane organelles? You can see that there are two subgroups, single membrane organelles, which has only one membrane in the structure, for example, endoplasmic reticulum, Golgi apparatus, lysosomes and peroxisomes. Double membrane organelles includes mitochondria and plastids. Non-membrane organelles group includes ribosomes and different components of cytoskeleton. In detail, we will be talk about some later. So, endoplasmic reticulum. The endoplasmic reticulum is a type of organelle found in eukaryotic cells that forms an interconnected network of platinate membrane-enclosed sacs or tube-like structure known as cisterna. The membrane of endoplasmic reticulum are continuous within the outer nuclear membrane. Classification. On the base of the structure of endoplasmic reticulum, there are two types of endoplasmic reticulum. Root endoplasmic reticulum, called root because has ribosomes on its surfaces, and smooth endoplasmic reticulum without ribosomes. So, root endoplasmic reticulum has ribosomes, and smooth endoplasmic reticulum has no ribosomes. The main function: translation and folding of new proteins. This is the main function of the root endoplasmic reticulum. And I remind you, what is a translation? Translation is the process in which ribosomes in cytoplasm or endoplasmic reticulum synthesize proteins after the process transcription of DNA to RNA in the cell nucleus. So it is a part of the biosynthesis of the proteins. And what is the folding? We said that the function is the folding of the proteins. For folding, it is the physical process by which a polypeptide folds into its characteristic and functional three-dimensional structure from a random coil. And main function of the smooth endoplasmic reticulum is expression of lipids. The next single membrane organelle is Golgi apparatus. The Golgi apparatus consists of several membrane-bounded cisternae, or another name saccules, arranged in a stack and positioned and held in place by microtubules. Structure: cis phase, located here. Cis phase nearest to roof endoplasmic reticulum, and trans phase farthest from roof endoplasmic reticulum. Functions: sorting, baking, processing, and modification of the proteins. Also, Golgi apparatus is the workplace where lysosomes are synthesized. So, now let's talk about lysosomes. Lysosomes are dense membrane-bound organelles of diverse shape and size that function to degrade material. They include enzymes, which called acid hydrolases, such as proteases, lipases, glycosidases. I remind you what it means acid hydrolases. Acid hydrolases are enzymes which disintegrate materials with help of hydrolysis reaction. And these enzymes work in acid environment, so it's called acid. Classification of the lysosomes. All early endosomes or lysosomes include inactive enzymes. Late endosomes play a key role in various lysosomal pathways. Residual bodies, or another name third lysosomes, are lysosomes of any type that have expanded their capacity to degrade material. And another classification, which based on type of digestive material. There are autophagy lysosome, is lysosome with digestive organ components of the cells, for example, damaged or aged. And heterolysosomes, lysosomes with digestive foreign particles which enter the cells procedurously. And let's talk about another type of organelles, single-member organelles, peroxisomes. Peroxisomes, or another name, microbodies, are membrane-bound spherical or ovoid organelles. Peroxisomes contain a crystalline core consisting of urate oxidase. Peroxisomes are similar to lysosomes, but unlike them. They do not contain acid hydrolase but oxidase. Oxidase are enzymes that break down substances by an oxidation reaction. That means that the compounds with the oxygen. The main function: the oxidation of long-chain fatty acids, detoxification of substances which exceptionally harmful to the cell. And now let's talk about double membrane organs, and we said that this type of organelles includes two main organelles. It is a mitochondria and plastids. So, mitochondria are rod-shaped organelles that are 0.2 micrometers wide and up to 7 micrometers long. They possess an outer and an inner membrane, and the inner membranes indigenates to form crystal. there are such parts of the mitochondria which called matrix and intermembrane space. The outer membrane also contains enzymes involved in such diverse activities as the elongation of fatty acids, oxidation of epinephrine, and the degradation of tryptophan. The mitochondrial intermembrane space is the space between the outer membrane and the inner membrane. It is also known as perimitochondrial space. Because the outer membrane is freely permeable to small molecules, the concentration of the small molecules such as ions and sugars in the intermembrane space is the same as in the cytosol. However, large proteins must have a specific signaling sequences to be transported across the outer membrane, so the protein composition of this space is different from the protein composition of the cytosol. One protein that is located to the intermembrane space in this way is cytochrome C. The inner mitochondrial membrane contains proteins with three types of function. Those that perform the electron transport chain redox reaction. Second, ATF synthetase, which generates ATP in the matrix, and the specific transport protein that regulate metabolite passage into and out of the mitochondrial matrix. The inner mitochondrial membrane is compartmentalization into numerous crista, which expands the surface area of the inner mitochondrial membrane, enhancing its ability to produce ATF. You know that the main function of the mitochondria is producing of the energy. In addition to supplying cellular energy, mitochondria are involved in other tasks such as signaling, cellular differentiation, and cell death, as well as maintain control of the cell cycle and cell growth. And let's talk some words about the matrix of the mitochondria. The matrix is a space enclosed by the inner membrane. The matrix is important in the production of ATP, with the aid of the ATP synthetase contained in the inner membrane of the mitochondria. The matrix contains a highly concentrated mixture of hundreds of enzymes, special mitochondrial ribosomes, tRNA, and several copies of mitochondrial DNA genome. Of the enzymes, the major functions include oxidation of pyruvate and fatty acid and the citric acid cycle. The DNA molecules are packed into nucleated by proteins. Mitochondria have their own genetic material and the machinery to manufacture their own RNAs and proteins. We talk about this function on the previous slide. So the main function, we said that it is a producer of the energy, heat production, storage of calcium ions, pyruvate and citric acid cycle, which occur in the matrix of the mitochondria. And we talked about the electron transport chain. Now let's talk some words about plastid. The plastid found only in the cells of the plants, algae, and some other eukaryotic organisms, but not in the animal cell. Plastids are the site of manufacture and storage of the important chemical compounds used by the cell of autotrophic eukaryotes. They often contain pigments used in photosynthesis and the types of pigments in a plastic determine the cell color. They have a common evolutionary origin and possesses a double-stranded DNA molecule that is circular like that of prokaryotic cells. And another and now we will talk about non-membrane organelles. Non-membrane organelles, we said that it is a ribosomes and component of cytoskeleton. So, ribosomes are non-membranous organelles that are 12 nanometers wide and 25 nanometers long and consist of a small and large subunits. So look at the picture, small and large subunit of the ribosomes. The two subunits are composed of several types of ribosomal ribonucleic acid or RNAs and numerous proteins. Ribosomes may be free in the cytosol or bound to membranes or the raw endoplasmic reticulum. We said so in this situation endoplasmic reticulum would be called roof endoplasmic reticulum, or the cytoplasmic surfaces of the outer nuclear membrane. A polyribosome or polysome is a cluster of ribosomes along a single strand of messenger mRNA, where every ribosome is concurrently engaged in protein synthesis. The main function of the ribosomes are the sites where mRNA is translated into protein. So ribosomes take a part in biosynthesis of the protein. And now let's talk about another type of organism which have not open membrane and it is called cytoskeleton. Cytoskeleton is a structural framework within the cytosol. The main function of the cytoskeleton functions are maintaining cell shape, stabilizing cell attachments, facilitating endocytosis and exocytosis, and promoting cell movement. So, there are different types of components of cytoskeleton. First, it is the microtubules. Microtubules are most commonly comprising 30 protofilaments that in turn are polymers of alpha and beta tubulin. Two monomers of alpha and beta tubulin are formed tubulin dimers. They have a very dynamic behavior, binding GTP for polymerization. They are commonly organized by the centrosome. In non-triplet sets, star-shaped, they form the centrioles, and in non-doublets, oriented about two additional microtubules, wheel-shaped, they form cilia and flagella. And the microtubules consist of proteins tubulin, we said, and in diameters 25 nanometers. The next type of cytoskeleton called actin filaments, or in other words microfilaments. This type of cytoskeleton in diameter 7 nanometers and global actin monomers G-actin, look at the picture, it looks like that, linked into double helix, which called F-actin or fibrillar actin. G-actin it means globular actin and F-actin it means fibrillar actin. Microfilaments are composed of linear polymers of G-actin proteins and generate force when the growing end of the filament pushes against a barrier, such as the cell membrane. They also act as track for the movement of niacin molecules that affix to the microfilaments and walk along them. In general, the major components of proteins of microfilaments are actin. The G-actin monomer combines to form a polymer which continues to form the microfilaments, or in other words, actin filaments. This subunit then assembles into chains that intertwine into what are called F-actin chains. Niacin motoring along F-actin filament generates contractile forces in so-called actin-niacin fibers, both in muscle as well as most non-muscle cell types. And the last type of components of cytoskeleton is the intermediate filaments. Intermediate filaments are in diameter 8 to 10 nanometers. These filaments are more stable, strongly bound than microfilaments and heterogeneous constituents of the cytoskeleton. Intermediate filaments organize the internal three-dimensional structure of the cell, adhering organelles and serving as structural components of the nuclear lamina. In combination with proteins and desmosomes, the intermediate filaments form cell-cell connection and unhole the cell matrix junctions that are used in messaging between cells as well as vital function of the cell. These connections allow the cell to communicate with the desmosome of multiple cells to adjust structures of the tissue based on signals from the cell's environment. And our last question on lecture today, it is inclusion. With that, I remind you that the cytoplasmic apparatus includes cytoplasm, we talk about it, organelles or organoids, and the last part of the cytoplasmic apparatus, it is the inclusion. What's mean inclusion? Inclusion are accumulation of material that is not metabolically active. They are usually present in the cytosol only temporarily. So, the main differences from organelles is that inclusions are only temporarily, but organelle it is a constitute components of the cell. So, let's see. Classification. There are different types of inclusion. The first it is a trophic inclusion. The inclusion which help to nutrition to the cell. For example, glycogen or some lipid droplets. So look at the picture. There are electronic microphotograms in which you can see the lipid drops in the cell, the trophic inclusion. Another type of inclusion it is a pigmentary inclusion. It is a different type of pigments. For example, the main pigment of the skin it is a melanin will give the skin their color. The next type it is secretory inclusion. The classical example is hormones. Hormones, the substances which must secrete from the cell. And another type of inclusion is the excretory inclusion. For example, urea. And very interesting type of inclusion, it is a random inclusion. It is substances uh, which not have to in the cells, but sometimes we found it in the cell. For example, it is some inclusions such as soot and dust. So, our lecture is over. Next lecture will be about structure of plasma membrane, about membrane transport. Thank you for your attention, and we will see with you on the next week.